In this tutorial, we will talk about the OmicsOffice SeqSolve application. SeqSolve is an application for NGS analysis that runs on the TIBCO Spotfire platform. In order to analyze an NGS experiment using SeqSolve, we need to load it using the OmicsOffice SeqSolve option in the Open From menu. This will present us with a dialog box where we can choose between two different workflows, one for ChIP-seq and one for RNA-seq. In this tutorial, we will focus on RNA-seq. We also have the option to choose between two modes of execution, local and remote. For this demonstration, we will use the local option. Once we've chosen these options, we proceed to a second menu where we will configure all the settings for the current analysis. First, we select the input files. These input files should contain reads aligned to a reference genome. The input file types allowed for these alignments are BAM, BED, or GFF. In this case, we will use the BAM files and we can use the file type filter to display only these files. Once the input files have been selected, we will choose the annotation that will be used for the analysis. It is very important that this annotation corresponds to the genome version on which the alignment was performed. After the alignment files have been selected, we can add additional information regarding the experimental settings using the Add Information button. This allows us to define the experimental design directly in the window it presents us with, or allows us to load a predefined file containing the experimental design settings. We will do the latter for this tutorial. The format of the file with the experimental settings is a simple tabular format containing each of the files, the sample name and the group to which it will be assigned. Finally, we will choose the predefined workflow we want to run. Alternatively, we can define a new one in case the available ones do not suit our needs. We will choose the complete workflow in this case. Now we have everything configured and we just need to click and go in order to execute our analysis, which will take slightly longer to run than the 2 minutes it took to set it up. If we had chosen to run SeqSolve server version, the computation would be sent to the server and we will be notified by email when it is completed. The resulting analysis will consist of several tabs with different results in each of them, which I will briefly summarize. First, we have a set of quality control tabs which include a general evaluation of the reads and mapping, distribution of the reads and the different features, and along the genome. Read counts for each of the genes and distribution of reads in exons and feature boundaries, such as transcription start and end or splice junctions. Gene and transcript expression levels in RPKM are reported, as well as information on splicing and unannotated transfrag expression. Once this initial analysis is run, a variety of tools for further analyzing the data are available from the Tools menu. We will have a more detailed look at these in subsequent tutorials.